and I welcome everyone for today's Matter to Life lecture series, a lecture series where we introduce the faculty of our Matter to Life Max Planck Schools program. And one of the beauties of the program is that our fellows are distributed all over Germany, as you can see here on the map. So we have 14 different locations where our fellows are. And today it's my pleasure to have Markus Müller with me, who is one of our professors at the University in Göttingen. And as usual, we like to get to know our faculty a little better before we dive into their current research. And for this, we have two questions prepared for you, Markus. And I would start with the first one, where I want to know from you, can you tell us about your current role at the Max Planck School Matter to Life and how your work contributes to the broader Matter to Life field?
Okay, hello, everybody. Thanks, Jan, for the nice introduction. So, I mean, my contribution to the Matter of Life School is basically threefold. So, on the one hand, I do teaching. I'm a theoretical physicist, so I offer the perspective of computer simulations and theoretical physics. I offer courses which are part of the physics curriculum in Göttingen as elective courses to Matter to Life, such as computational physics or soft matter and polymer physics. Then, I mean, research-wise, I do soft matter and biological physics. And in my talk, I have one extra slide on that to introduce different topics because the topic of the talk is fairly specific. And so, as I said, we do polymer materials and biological membranes. And with this, I think we are right at the intersection between synthetic and biological matter. You can, of course, also ask what does Matter to Life does for my research. I think this is also very important and a motivation to participate here. And it's a great opportunity to collaborate and to get inspirations for projects and motivations for topics. So I had really excellent experience with these lab rotations, both with the Matter to Life students as well as colleagues in Göttingen, like Stefan Klump or David Zwicker or Tim Saldet, as well as Jo Bockhöfen and Erwin Frey in Munich. And so these contacts, which were established through Matter to Life, persist even over the lab rotation away. So that is very good for us.
Great.
And then, I mean, the third one is very little. I'm a little bit involved in this administration by being part of the selection committee and the Studiengangskommission. Not quite sure what the English word for that is, probably program committee or something like that. That I do too, participate in.
Thank you very much. We're very happy to have you with us. And the second question we always ask our faculty members are, what are some of the most significant challenges you've encountered in your career so far, and how did you address them?
Well, I mean, challenges, there are many, right? So I have selected sort of two, one general and one specific for my research. So the general challenge is how to choose a good research topic. That's always an important choice, right? And I mean, it's of course a little bit of luck and serendipity to find a nice, interesting question, which is just about enough challenging that you can solve it. But what is also important is, of course, hard work and knowing the literature, very important, because especially maybe in theory, nothing is more frustrating than sort of working on a topic and then figuring out somebody else did it 15 years ago and you didn't find it. So I think that's very important. And also it helps to engage in discussion with colleagues. I mean, even on topics which are only remotely connected to that, because if you then ask yourself, how would this strategy work for my problem, you often generate new ideas and new projects. That's very good. And then the more specific challenge to my research field, so we do large-scale computer simulations. We, of course, use standard programs, but we also have programs which we have written on our own. And so maintaining these programs and adapting them to new computing paradigms or hardware like the exascale computer Jupyter now in Jülich, that is a challenge, especially if people come and go in the research group. And so it is important to have some people overlapping, which have interest in programming and program design and algorithms, in addition to the science. And often this is not sort of something different, because if you develop new methods, then this opens also new avenues for simulations which were not feasible before. Also, I mean, I had very good experiences with Hiwis. There are some cracks who really like to program, so that helps too. And if time permits, I also do a little bit of programming myself in order not to lose connection to that computational aspect.
Great, thank you so much. And now we're very much looking forward to dive into your current ongoing research. And with this, I'll hand over to you, and the stage is yours.
Okay, thanks. Okay, okay. So, I mean, what we are doing is soft matter and biophysics. For the talk, I have selected sort of a quite specific topic, which is about one paper. But I mean, in order to give you a little bit broader overview, we do both synthetic self-assembly as well as biophysics. So we have a lot of activity in self-assembly of block copolymers, amphiphilic molecules. You see here an example where you form membranes, not biomembranes, but filtration membranes, by one material where you have a selective top layer, which forms hexagonal pores, which are uniform in size. And then down there, you get macroporous material, which sort of endows the thin layer here with mechanical stability. So if you filter something through with a pressure gradient, the membrane doesn't rupture. And this is one material, and there's a certain processing where you first evaporate the solvent, and then you exchange the remaining solvent with a non-solvent, typical water, in order to get this non-equilibrium structure. Then we look at reaction-driven self-assembly, and I had this very excellent Matter to Life PhD student, Gregor Heffner, who did that. So where these molecules can change their identity from water-soluble to amphiphilic, and then they self-assemble in those vesicles here. And you can see that they also create, in this case, this pore, which sort of can propel them forward. And they create this reaction, create concentration gradient, and you get diffusiophoresis of these vesicles or these droplets in their own concentration gradient. Another student, Lucia Wesenberg, looks at the absorption of vesicles on surfaces, so on much larger length scales where you can treat the membrane just as an elastic sheet. And then we have a lot of activity in non-equilibrium structure formation, like this process-directed self-assembly, where you try to devise processing strategies where you change the free energy landscape and the system runs into the nearest metastable state. And by that, you can create structures. The membrane structure on the left-hand side would be one of those examples, which do not occur in the equilibrium phase diagram, but which have interesting properties. Okay, and you also see these symbols here. So we do a lot of computing. We use large computers like the computers in Jülich with many thousands of GPUs. Let me start with a proper talk from fusion to fission, insights from modeling. This is work by Russell Spencer, Yulia Smirnova, and Alireza Soleimani in my group. And the question is as follows. So you are at the synapse. You have here the presynapse, and you load vesicles, synaptic vesicles, by these neurotransmitters. And then upon stimuli, these neurotransmitters are released in this synaptic cleft, and then trigger the reaction on the postsynapse. And this is done very frequently, and so you have to recycle those synaptic vesicles. And there are two processes. So one process is the synaptic vesicle comes down to the plasma membrane of the presynapse. There are snares which sort of dock the vesicles to the plasma membrane. Upon calcium trigger, they do a conformational change exert forces on this and put the membrane under tension. Then you form this hydrophobic connection between the synaptic vesicle and the membrane, which is called the stalk. It's a hydrophobic bridge. People have researched a lot about what is the free energy barrier to the formation of this bridge. This bridge expands into a hemifusion diaphragm, where you have a single membrane which connects the plasma membrane and the vesicle membrane. And then you nucleate a pore inside of this hemifusion diaphragm. So this here, you will see more of those pictures, is a top view from here. You see here the membrane of the synaptic vesicle. Here you see the plasma membrane. This is the connection. Inside of this connection, you have a single membrane, and then inside of the single membrane, you nucleate a pore. The pore forms at the rim of this hemifusion diaphragm, and therefore it's called a rim pore. And through this pore, the neurotransmitters can be released. And then you have to revert this in. And ideally, this endocytosis, the reversion process, just follows the same steps here. But in order to do that, you would have to arrest the study in this structure here with a rim pore. And that is what the talk is about. So the question is, how can you if the vesicle comes down, it fuses with a plasma membrane, releases the neurotransmitter, how you can reverse this? And there are two mechanisms. The classical mechanism is clathrin-mediated endocytosis. So what happens is you form this structure here where you have a fusion pore, which connects the synaptic vesicle and the plasma membrane. And then basically the synaptic vesicle integrates into the plasma membrane, as illustrated in this movie, if it works. Maybe I click on the wrong thing. Okay, so it comes down, it fuses, forms a stalk, you porate it, and now the vesicle sort of integrates into the plasma membrane. And then later, and typically also at a different position, you butt off the vesicle to recycle it using clathrin as scaffolding proteins. The alternative to that is that the first stage is the same. You form the stalk, this hemifusion diaphragm, you create a pore. Through the pore, the neurotransmitter are released. But then instead of collapsing into the plasma membrane, you just revert those steps. And this is called the kiss and run pathway. And the molecular pathway is incompletely known. However, it seems to be important because, I mean, these synaptic vesicles are complex. The picture is on the next slide. So they have a very specific composition of proteins, and they have a very uniform size. And in order to contain this morphological uniformity and the protein composition over repetitive rounds of this neurotransmitter release and exo and endocytosis, it would be nice if you don't lose the identity of the synaptic vesicles as you do when you do this clathrin-mediated mechanism. So here the hypothesis is that you arrest the fusion process in this state here. So this is the synaptic vesicle, the plasma membrane, the hemifusion diaphragm, the single membrane that connects the vesicle and the plasma membrane. Then you nucleate a pore. It's heterogeneous nucleation. It's better to nucleate this at the rim of the plasma membrane down here. And through this pore, you can release the neurotransmitters, but this should remain like this, and it should not open to a full fusion pore, because then basically this is like a parachute which just integrates into the plasma membrane. So you want to arrest the process in this rim pore state such that it doesn't collapse, and this is the extended hemifusion state, which is a intermediate in this kiss and run mechanism. What you see here is also two types of simulations which we use. And so we tried this first by particle-based simulations. So we use Martini, the coarse-grained force field, where you have specific models for different lipids, and GROMACS, molecular dynamics program. You build a membrane, you build this hemifusion diaphragm with a pore, and then you let it run. And what happens is that either the pore shrinks to zero or it expands to a full fusion pore. So that was sort of disappointing at the beginning. So there seems to be these two stages. I mean, basically it collapses or it opens to a full fusion pore, but this rim pore does not seem to be a metastable state, and therefore it seems to be unlikely that you can arrest the system in this state and use this as an intermediate from which you can then revert the exocytosis to the endocytosis. Of course, this is not the full story, so I should mention this picture here of a synaptic vesicle. It doesn't contain only lipids, but it is crowded by proteins. So there are additional interactions and constraints acting on the synaptic vesicle. We also tried this and to understand this by looking at the free energy of the system. And so what you see here is the minimum free energy path. So you can follow sort of the free energy of a system as it sort of expands this rim pore into the full fusion pore. So here you see the free energy. The typical scale of the free energy is the bending rigidity. On the x-axis, you have a reaction coordinate, which says how far you progress along this path, where you open this rim pore. And you see here data for different radii of the rim pore. So the black curve is if it is just an infinitely large rim pore. This is a still large, and this is a smaller rim pore. And the typical free energy landscape has two barriers. So at very small, initially in the path, you have a barrier here where you start to nucleate this rim pore at the rim of the hemifusion diaphragm. And then sort of the rim pore relaxes into this metastable state, but it's very small. So basically, that's not good enough for neurotransmitter release. It's called a pre-pore, and this has been also observed in membrane pores in a normal membrane, not with this hemifusion diaphragm around. And it basically means the pore wants to close, but you have these head groups, and so the head groups just touch each other, and you don't make the transition to an intact membrane, but you basically close the pore with the head groups. This is this pre-pore stage. And then you can expand the pre-pore, and you have a free energy barrier associated with this. It's a critical pore, and if you surpass a critical pore size, then the rim pore grows and fills the full hemifusion diaphragm. Okay, so that is the result of the simulations and of self-consistent field theory, and I will explain what self-consistent field theory is later on. It's a very nice method to obtain free energies. And so the finding is that in the canonical ensemble, you just fix the number of particles in your simulation box. This rim pore in a hemifusion diaphragm is not metastable. You can say, well, why should it be? And why we first initially thought why it is metastable is that if you have a single membrane in the canonical ensemble, then a pore in a single membrane is metastable. And the size of the pore and the membrane tension are simply related by the Laplace pressure. So the line tension of the pore divided by the radius of the pore is equal to the membrane tension. It's mechanical stability of the size of the pore, right? And now here in this case, I mean, even you're in the canonical ensemble, it's not stable. And the reason is because the size of the hemifusion diaphragm can vary. And thereby, as it varies, it basically acts as a lipid reservoir. And so the pore in the hemifusion diaphragm looks much more like a pore in a membrane at constant tension. And that indeed shows the same. There's a critical pore size. And so if you want to have this rim pore in the hemifusion diaphragm as an intermediate, you somehow have to make it metastable and to make it longer lived. So this is the question we want to tackle, how to make this metastable, and we used 3 techniques. So one technique is just analytical theory. We write down the free energy of the rim pore in a phenomenological model. This is shown here, and this is what we do first in order to just get an overview of what is the free energy landscape of the system. Then we use self-consistent field theory, which accounts for the lipid architecture and the self-assembly, and additionally, it can easily calculate the free energy. And then we verify the findings by coarse-grained particle-based simulations using the Martini model and the GROMACS program. Okay, let me start with the phenomenological model. I mean, in principle, it's very simple. You describe the system by this geometry. The red circle is the hemifusion diaphragm. It has a certain radius. Then in the hemifusion diaphragm, you have a pore. This is this pore here. This is the edge of the pore, and the edge of the pore looks like the end of a membrane, so it has this line tension of the edge. Then you have this rim here, and this rim here is like a fusion pore. So the membrane just bends around, and the red one is this hemifusion diaphragm, which is this line here. You write down the free energy as the line tensions of these 3 structures times their lengths. And then you have the area constraint because you're in the canonical ensemble. The total membrane area is conserved. And you have the membrane tension, which is this Lagrange multiplier here, which fixes the area. Okay, in principle, it's very easy. Due to the geometry, it's a little bit complicated, so it's a long formula, but this is what it is. The free energy is the red line, length of the line times the tension of this 3 bilayer junction, the length of the blue line times the line tension of the edge, the length of the green line times the line tension of this fusion pore, and then you have membrane tension, which controls the membrane area. Okay, if you do this, then basically you can minimize the free energy with respect to the geometrical parameters of this. And if you minimize this with respect to the radio of this edge of the fusion pore and of the hemifusion diaphragm, you basically get Laplace equations, as you would expect. So you get that the line tension of the edge here, the blue one, divided by the radius of this edge is equal to the membrane tension. If you have the fusion pore, there's a factor of 2 because there are 2 membranes attached to it. And if you have this hemifusion diaphragm, then you have 2 membranes here, 1 membrane here, so you get 1 tension, and you get the Laplace equation as well. So the tension is equal to the line tension of the red line divided by its radius. So that's good. And then you can sort of see how the free energy looks like as a function of these radii. And you see that it's convex in the radius of the pore and the edge, but it's concave, so it has a maximum in the size of the hemifusion diaphragm, right? And so you see that the curvature at this point, which minimizes the free energy, is not uniform. You have positive and negative curvature, and therefore this is a saddle point, as you observed in the simulations. And you also see what is responsible that it's a saddle point and not a metastable local minima. It's the radius of this hemifusion diaphragm. So if you change the radius of the hemifusion diaphragm, the free energy goes through a maximum. And this is exactly what we saw in the simulations. So either you are on the small side, then it collapses, or you're too large, and then it opens up to a full fusion pore. The good thing is you can sort of look at the geometry of this rim pore as a function of the different parameters. And so in the Hamiltonian, basically, you have the three line tensions. From the three line tensions, you can form two-dimensionless ratios, the line tension of the edge versus the line tension of the hemifusion diaphragm and the line tension of the pore, divided by the line tension of the hemifusion diaphragm. And then in this space, you can see where it is possible to have these critical rim pores, and it is inside of this square here. If you're in the middle of the square, then you get a geometry which is similar to what I've drawn. And then if you go to the edges of the square, like if you go to this end here, then you see that the pore becomes very large and tries to butt off the rim pore. Likewise, if you go here to number 5, then you see that the pore tries to detach from the hemifusion diaphragm. So it goes into the middle of the hemifusion diaphragm and is no longer a rim pore. So that's nice. So you can get a feeling, what is the geometry, where is the rim pore, under which circumstances it is stable. You can also associate with the line tensions of the edge and the hemifusion diaphragm and the pore certain lipids, and then you see that different lipids are at different positions in this phase diagram of critical pores, and they correspond to different geometries. Okay, so in this sense, the phenomenological model can explain the findings of the simulations. It sees that there are critical pores in a certain range of parameters for lipid architecture, and they're critical and they're not metastable. And it also sort of identifies the culprit, why they are metastable, not metastable, but critical, because the hemifusion diaphragm can change its size. In this one, it is concave. Then we turn to the second method, and that's self-consistent field theory. And in self-consistent field theory, you do use amphiphilic molecules in order to assemble the membrane and the hemifusion diaphragm and so on. And the way it works is that you look at a molecule which is in a field which represents the interaction of the molecule with its environment. And this is the field V, W. And if you have this field, you can look at the statistics, the density distribution of those lipids in the field. From the density distribution, you can calculate where are the hydrophilic and hydrophobic groups in a function of space. Once you have this, you can say what are the fields of the environment, and you circle this around, and therefore it's called self-consistent field theory until the fields and the densities are related by self-consistent equations. If you do this, these amphiphilic molecules assemble into membranes, and here's the phase diagram as a function of the hydrophobic fraction and the membrane tension or chemical potential. And then you see that you will form spheres like micelles, you can form worm-like micelles, and you can form stable membrane flat sheets. And you can also relate the parameters of the model to real lipid systems by identifying the energy scale by the bending rigidity and the length scale by the thickness of the membrane. This works quite nice. Then you can calculate, also correlate the fraction of the hydrophobic units to the spontaneous curvature of the monolayer. That works as well. And you see that also the bending rigidity of the membrane is rather insensitive from the hydrophobic fraction, but it depends on the incompatibility between hydrophilic and hydrophobic segments, which is characterized by this chi parameter here. You can then, of course, use this in order to calculate the line tensions of the different structures. as a function of incompatibility and hydrophobic volume fraction. And it's sort of easy to qualitatively understand the trend. So if I make the hydrophobic fraction large or the head group small, then this type of curvature at the edge becomes more expensive, and the opposite type of curvature of the head group region becomes more favorable if I go to this part here. So meaning that if I increase the hydrophobic fraction, the pore edge becomes more expensive, and the hemifusion diaphragm becomes less expensive if I increase it. Okay, with this, then I can sort of look at really the phase diagram. And what is shown here is the configuration of this rim pore as a function of incompatibility between the hydrophilic hydrophobic units and this hydrophobic fraction, or the spontaneous curvature of the monolayers, if you want. And you can see 3D regions. So you see this region here where the rim pore really tries to expand. And this is if the line tension of the pore and the edge, this is the edge and this is the fusion pore, if the sum of those two is less than the line tension of the hemifusion diaphragm, then basically the thing becomes unstable and you rupture the hemifusion diaphragm. This is in this region here. In the opposite case, the hemifusion diaphragm is stable. You don't form a rim pore. And then you have these metastable states here with little pores, and these are the pre-pores you saw in the molecular dynamic simulations, where basically the pore wants to shrink, but it doesn't manage to rearrange the head groups, and you get these very small pores which are filled by the head groups of the lipids, which you would see in experiments by flickering pores where you have a little current going through. Okay, now you can do this now with the rim pore in the canonical ensemble. And what you see then is that if you have a large hemifusion diaphragm, then the tension is small. And if the tension is small, basically the rim pore is subcritical. It wants to shrink, and it shrinks to this metastable pre-pore. If you make the hemifusion diaphragm smaller, then the tension is higher because of this Laplace equation, which says the tension is equal to the line tension of the hemifusion diaphragm divided by its radius. And then you see that the rim pore, the pre-rim pore, sort of grows a little bit. And if you do it too much, if you put the membrane under more tension, then it suddenly opens and forms the full fusion pore. Okay, so that's the bad case. But then, I mean, we learned from the phenomenological model that you have to, in order to have a metastable rim pore, the radius of the hemifusion diaphragm must not change. And you could imagine that around this fusion site, you assemble, for example, a collar of proteins like dynamin, which are also implicated in severing off and tubes and in the fission, which simply constrains the radius. And this is shown here in this graph. Let me see that I get my mouse back to the screen. So this one here is where you have an equilibrium-sized hemifusion diaphragm, where the tension of the membrane and the line tension and the radius are all equilibrated. And now you try to squeeze the hemifusion diaphragm. By squeezing it, you increase the tension because you're in the canonical ensemble. So the area where you have two membranes is larger than the area where you have one membrane. So the tension goes up, and you get this very small pre-pore. And as you start doing this, this pre-pore increases and increases, and you can continuously tune this metastable rim pore in size by simply changing the radius of this hemifusion diaphragm. And that's the metastable state that we would like to have. And you understand that there are two constraints necessary for that. So there's a chemical constraint, which is given by the canonical ensemble, NBT, where you basically don't exchange, the number of lipids is conserved, so you don't exchange lipids with the environments. And in addition to that, you need this mechanical constraint, which prevents the hemifusion diaphragm from changing its radius. And that could be done by proteins, for example. There's also another way of doing that, and this is simply by suppressing lipid flip-flop. This also constrains the size of the hemifusion diaphragm. And this is also what is naturally operating in molecular dynamics simulations. So what you do is, here is the hemifusion diaphragm. These are the two membranes. This is the diaphragm. There's another two membranes. And what you can do is you prevent that lipid flip-flop from the upper leaflet to the inner leaflet here. And in self-consistent field theory, you do this basically by having two different head groups which repel each other but have otherwise completely identical properties, and then they don't mix. So then the number of lipids in this leaflet here is conserved, and the number of lipids there dictates the size of the hemifusion diaphragm, right? So this is the radius, I mean, basically, and you can't change this. And if you do this, then this is the mechanical constraint you need for this position here not to move. And so you can look then how the system evolves as this pore forms. This is, again, this minimum free energy path. It's something nice in, let me start the movie, nice in the self-consistent field theory, because you get the sequence of configurations how such a pore would evolve. And of course, you get the free energy scale of it. And this is now the free energy as a function of the progression, how this pore starts. Before that, when you do not have a constraint or suppression of the flip-flop, you had this green curve here, which had two maxima, the formation of the pre-pore, and then this critical pore size. And now what you do is you simply avoid the flip-flop, and then you get the black, blue, and red curve, depending on how many lipids you have in this monolayer here, in the outer monolayer. And you see that you get first a peak in the free energy profile. That's the formation of this pore, this one here. But then, unlike the green curve where you had a maximum, here you have a minimum. And that minimum comes from it's a metastable state, and that's the metastable rim pore. And it's metastable because you have this constraint. So this constraint converts basically the critical pore into a metastable pore. Okay, and you see the same thing in the self-consistent field theory and the simulations. Again, here you have these different numbers of lipids in the outer leaflets. And by that, you can control the size of this now metastable pore. And indeed, you see also a metastable pore in the MD simulations. You can ask, of course, what is the difference between this situation here and the situation before? And the difference is that there's no lipid flip-flop. And in the previous simulations, you saw far away from this a little pore which connected the two leaflets of the membrane, and the lipids can go around, flip-flop this pore, and thereby you transfer lipids from one leaflet to another leaflet. And so if you do this in the particle-based simulations, what you see is here again the hemifusion diaphragm, the rim pore. If you don't have flip-flop, then the rim pore stays. This is a hemifusion diaphragm. This is a two bilayer region. This is a pore. And then if far away from it, you create a pore in a single membrane, and that allows lipids to go between one leaflet and another leaflet, then at this point here, the pore shrinks to zero, right? So it vanishes. So which means this is a critical state. It's not metastable any longer. And so you have sort of like a remote control, if you wish. If you don't have this pore far away, then this rim pore is metastable. And if you allow for lipid flip-flop by something, then it becomes critical and it either grows or it shrinks, right? Okay, and with this, I'm at the end of this talk. It had focus on a little specific problem, namely this transition state of the kiss and run mechanism, which is required for this fast and sustained synaptic release, where you recycle the synaptic vesicles rapidly and avoid that they collapse into the plasma membrane. This rim pore is a putative intermediate because the vesicle identity remains the same. We have identified a suitable region of line tensions where this rim pore can exist, and we can relate this line tensions to specific lipids or lipid mixtures, and then say what is the geometry of this rim pore. And I think the important part, or the one which was surprising for me, was that these rim pores weren't metastable if I simply fix the number of lipids. That's different from pores in a normal flat membrane, but you need two constraints. One is the chemical constraint that you do not exchange lipids. And this, if you think of this picture of the synaptic vesicles, which is crowded by many, many proteins, then you can imagine that diffusion is slow. And so diffusion of lipids and lipid exchange between this fusion site and the rest of the membrane can be slowed down. But this alone is not sufficient. You also have to constrain the size of this hemifusion diaphragm. And there are two ways we suggested this can be done. One way is to put a protein ring around it, and that could be Dynamin. And another way that could occur is if you suppress this lipid flip-flop, and lipid flip-flop is a slow process in real membranes. So you can basically then switch from this metastable pore to one which reverts to the stalk by either having a subcritical rim pore, and if you release the constraint, then it just shrinks to zero, and then a stalk forms and you reverse the fusion pathway. And this can be done either by disassembling the constraint of the proteins, or just wait long enough that you have lipid flip-flop, and that will also release the constraint. Again, I'd like to thank the people who were involved. I want to give a little outlook, just because this is a nice movie. So this is this pore, and we have now a protein ring which shrinks it together, and you see that it sort of squeezes off this tube. It's a little bit different geometry, but this is done by self-consistent field theory, and you can look at the free energy profile of this, and you see that the proteins really have a strong influence on the barrier, and this also depends on the interaction between the protein and the lipids, and this is what we are going for thereafter. Let me just show the people who were involved. Julia Smirnova and Alireza did the MD simulations with Romax, and Russell did the SCFT calculations and the analytic calculation. And with this, I thank you for your attention and be happy to take questions.
Hey, thank you very much for this great presentation and the insights in your current ongoing research. And for everyone in the audience, I now open the stage for the discussion. So please use the question and answer bubble on the bottom of Zoom in the toolbar, and just type in your question, and I will read it out to Markus, and he will gladly answer all the questions you might have. And there's already the first one, so I would start with this while others might be still typing. And this is, in this kiss and run model you showed, is there a possibility that not all the neurotransmitters will be released into the postsynaptic neuron? In other words, how does the time scale of this transient kissing and running compare to that of the neurotransmitter diffusion?
That's a good question. It depends how long the metastable rim pore stays open. And this depends on how long it takes to release the constraint. So if you would wait for a lipid flip-flop just to occur in order to release the constraint, that would be long. If you would have this dynamin collar, which you disassemble, or another protein collar, that could be faster. But it may be also not too bad if not all the neurotransmitters are released, because, I mean, this kiss and run mechanism is particularly suitable if you have a high frequency of this release, right? So you want to have, I mean, many pulses in a short time. And so maybe it's okay if you just release a few, then you close it, but it stays at the active zone of the synapse. And then if the next pulse comes, it does it again, right? So what I didn't explain is you have different vesicle pools in the synapse. You have this, let's say, storage pool, and then there are vesicles which are docked, and those ones then can release the neurotransmitters. If you want to have rapid firing, basically, right, then, I mean, it wouldn't be a disadvantage if not all neurotransmitters are released in one shot. But to this question, I mean, this is a theoretical suggestion, right? So I think experimentally the mechanism is unknown, and I mean, I should explain that there are also people who believe that it simply doesn't exist. I mean, even so, the hypothesis is a long time around. But I think it's and the difficulty in experiments is, of course, that the time and length scales of this release are difficult for experimental observation. So they can globally measure the rate of release. They can take snapshot pictures with cryo-EM. But I mean, really seeing the process is a challenge. And this is where these calculations can help. And if it is only in the sense that they discuss what is possible. I mean, the validation, of course, has to come from the experiment.
Question from my side, would that be also, if you have different neurotransmitters, is then the release time is probably a different one, or is it the same for all different kind of neurotransmitters?
Experimentally, I don't know. In the model, of course, if they have different diffusion coefficient, it's different. I mean, there's also another possibility how you could trigger the reversion from fusion to fission. And you could imagine that the neurotransmitter interacts with the membrane of the synaptic vesicle. And once it's sort of released, then the inside of the synaptic vesicle is a different one. That changes the membrane properties. It could change the tension, for example, and that in turn could also trigger the reversion.
And then also, I mean, if you would, I mean, there are multiple drugs that have influences on neurotransmitter release and so on, right? So this would also then influence, I assume, this behavior, no? Where am I going? This now is my
I mean,
a long time ago, sorry.
I'm not exactly sure about which drugs you're talking about, but I mean, sure. I mean, there's a chemical specificity to it, right? So, I mean, the membrane properties are not only dictated by the lipids in the membrane. There are also, of course, these proteins, and there's also the question: what is the solution around it, both within the synaptic vesicle, in the synaptic cleft, and within the presynapse. And these fluids, at least between the interior of the vesicle and the synaptic cleft, are sort of mixed, right? And this will change properties. And if there are drugs, they're included, or proteins which absorb onto the to the vesicle, that will change the physical properties of these vesicles.
Thank you. Maybe another question from my side also. How would you say now, when you look at your simulations and so on, now in the age of AI, how much would that affect simulations and what you can expect for the future? Is it getting then more precise or more?
I mean,
Yeah.
I think if you have, I mean, so if you want to use AI, you need data.
Yeah.
And you need tons of data. So if you have a model like this phenomenological model, then I think this is very well suited, and you can do high-dimensional parameter optimization. So now the phenomenological model was sort of simple. It had the geometry and three-line tension and the membrane tension. But you could imagine you have a very simple model. This has tons of parameters. And then you want to explore the behavior maybe by a little simulation of this model, and you can create fast, sufficient many data. That is good for, I mean, some optimization, active learning, machine learning, and things like that. And I think this is very good for finding parameters. If you think of the GROMAC simulations, I mean, they take a long time. And I mean, you will not be able to do 1,000 of those simulations. So I think for those, at least for the current state of computers, it becomes more difficult. Having said this, I should say, I mean, Jülich has now 6,000 plus GPUs of the newest generation. So this borderline is shifting continuously, right?
Okay, thank you. Then there's another question in the question and answer section. What's the link between the free energy and the functional availability of the protein for the next fusion event in the kiss and run mechanism?
The free energy and the functional availability. So, I mean, the free energy is, of course, strongly influenced by the proteins. So the idea of this kiss and run is, of course, that the proteins stay in the synaptic vesicle. And thereby, I mean, if I translate functional availability simply into there are the proteins at the right place, then kiss and run is very good for that. While if you think of the glycerin-mediated interaction, you basically have to, I mean, then the synaptic vesicle disintegrates into the membrane, and in fact, not even at the same position, but further away, it sort of butts off. And this means you have to rebuild the synaptic vesicle with all its sophisticated proteins and lipid composition from scratch. So in this sense, I think this kiss and run is very good because it retains the proteins and makes them available for the next step. You still have to fill it with new neurotransmitters, but I mean, at least the proteins would be there.
All right. Thank you very much. Since there's no further question, the question and answer bubble, I would say thank you very much for your presentation, for the insights in your current research, for the nice discussion. Yeah, and I'm looking forward to see you at one of our next events or somewhere else. And everyone else, have a great day. Enjoy the sun. Most likely, I mean, here at least it's very sunny. So wish you all a great afternoon and see you maybe at one of the next lecture series. Yeah, and with this, have a nice afternoon. And dear Markus, thank you very much.
Okay, thanks for having me and all the best to everybody.
Thank you. Bye.
Bye-bye.